All right, before we get started, I would just like to point out that even though it looks like him, this is not one of my cats. If I had done that to my cat, I would come in completely chewed up and scratched beyond belief. So what we're going to talk about are not only mechanisms of non-Mendelian genetics, but we're also going to talk about how environment affects the expression of genes. So first, before we can kind of go into this, let's talk about crossing over and recombination. And my bad, I changed this up so I could put this in your notes, but this chromosome is actually green in later slides. So let's say we have a pair of homologous chromosomes here. If there is no crossing over, no recombination, there are limited combinations of alleles that you can pass down. So let's say the pink chromosome has only dominant alleles for these 4 traits, for these 4 genes, and the gray, although it's going to end up being green later, chromosome only has recessive alleles for these 4 different traits. Without any kind of recombination, each gamete can only get one of these 2 possible combinations of traits. However, during crossing over, during meiosis 1, during prophase 1 and metaphase 1, you have crossing over occur where chromosomes can swap parts with each other. What this does is this produces different genetic combinations. And while we're looking at a super simple example here, just note that you can actually have multiple crossover events between the same 2 homologous chromosomes. So you could have a chromosome that by this model would end up being part pinkish purple, then part green, then some more pinkish purple, then more green, and so on. But we're just looking at one single crossing over event. So notice how the single crossing over event has kind of changed up the combination of traits that can be given to a gamete, to an offspring. Now, this is non-Mendelian inheritance because it doesn't follow the law of segregation and the law of independent assortment, because you can have genes that are inherited together. But we'll talk about that in a moment. So what we assume with this is that the probability of recombination is about equal at any point on this chromosome, although in reality, since all of this was first proposed, we found that there are some areas that are going to have lower recombination rates than others. So what this means is, in theory, each of these 2 recombination events is equally likely. And then what you can do is, in a particular genetic cross, you can calculate the frequency of recombinants versus parental types. So the parental types would be, for example, all dominant and all recessive. Anything besides these 2 would be recombinant offspring. And so basically the closer 2 chromosomes are physically located, the closer 2 genes are physically located on a chromosome, we would calculate a lower probability that you'll have a crossover event between them. So high probability you'll have a crossover event between genes A and B. Low probability you'll have a crossover event between genes C and D. So it's much more likely that you'll have like a differing combination of maybe, say, all capitals until you get to D, or all lowercase until you get to D. These are going to be rare recombinants. Because there's such a small distance between gene C and D, the probability of a recombination event happening between them is pretty low. So what we would say is these 2 genes tend to be linked in offspring, meaning if you inherited the dominant copy of gene C, it's highly likely you'll also get the dominant copy of gene D. And same thing with the recessives. So you can actually do some math with this, which we're not going to do, and you can calculate what's called the recombination frequency. And then back before we had a lot of complicated genetic technology, what people did is they started creating what they called linkage maps, or gene linkage maps. And they used this to kind of approximate where genes were located on a chromosome. So for example, a lot of this work was done with fruit flies, or Drosophila melanogaster. What they found is that when they looked at this, they had 4 groups of genes that tended to be linked with each other in some way. And then when they looked under a microscope and they looked at chromosomes in Drosophila, they found there were 4 chromosomes. So they were able to say, hey, I bet each of these groups of genes is located on a different chromosome. Nowadays, we have more accurate means to show this. So this would be a genetic map, which is basically based on recombination frequencies. We also have cytogenic maps, which are based on kind of relative location of different staining bands on a physical chromosome. And then we have kind of physical maps that are based on DNA sequences, since we've sequenced the genomes of multiple organisms. So you can go in and you can like physically determine, okay, this is where this gene is, this is where this gene is. Now, it's not as simple as, say, just looking at DNA. For example, when I was your age, they were just working on sequencing the human genome, and at first they thought we had 80,000 genes. But as they started to analyze the data that they were getting, by the time I was in college, the estimate had gone down to, oh, we probably have only 40,000 genes. And nowadays, the more commonly accepted answer is that we have about 20,000 genes. So it can take some time to distinguish what parts of DNA are coding for genes, coding for proteins, and which parts are not. But we'll talk about that more when we talk about regulating gene expression. So let's talk about some other types of inheritance that are not quite going along with Mendelian inheritance. We've actually already talked about some. Anything that's not straight up complete dominance is going to be non-Mendelian. So we have incomplete dominance, codominance, and also sex-linked traits. Those are all non-Mendelian forms of inheritance. But we got a couple of other things to look at. The reality in humans and a lot of organisms is that each individual trait is not necessarily linked with a specific gene. You can have one gene that has roles in multiple traits, and you can have one trait whose the phenotype is determined by a number of different genes. So here we see an example of, and this is not representing actual genes. This is all just kind of hypothetical, but different combinations of how 3 different alleles could contribute to pigment in skin color. So depending on the identity of the alleles that you have, it could give you variations in either very dark skin coloring or very light skin coloring. In reality, there's multiple genes that will interact, more than probably just these 3, and it's very hard to link conclusively most traits in humans to one specific gene. This is called polygenic inheritance. There's also DNA found outside of the nucleus in our cells, and in our cells that tends to be found in our mitochondria. But mitochondria are not inherited the same way that the rest of a cell is. Instead, while you get nuclear DNA, the DNA in your nucleus from every single one of your ancestors, as you see right here, you actually only get your mitochondrial DNA from your mom. Doesn't matter if you're male or female, every single one of you got your mitochondrial DNA and your mitochondria from your mother. So if you have any traits that are due to mutations in your mitochondrial DNA, then they will be passed from mother to offspring. And this is going to have some interesting kind of connotations when we get to evolution. So if we're looking at this, let's say we have 2 different cells that are going to give rise to gametes, and these are maternal and paternal, doesn't matter which one is which, but each of them has genetically distinct mitochondria as are represented by the different colors. So a sperm cell has very few mitochondria, and it's got very little cytoplasm. It's mostly a nucleus packaged with a tail. And the few mitochondria that it does have are there to just power the flagellum, so that way it's able to swim to the egg cell. On the other hand, a mature egg cell or oocyte contains a lot of mitochondria and a large amount of cytoplasm. Once fertilization happens, the zygote is only going to contain mitochondria coming from the mother. Basically, when a sperm cell fuses with an egg cell, the nuclear material from the sperm cell enters the egg cell. Those two nuclei fuse to form a new diploid nucleus, but not really any cytoplasm is going to come in from the sperm cell. So an egg cell is going to provide all of the mitochondria that an individual has, which means you get that only exclusively from your mother. So, for example, if you had a mutation, like I said, in a mitochondrial gene, that would only be passed from a mother. The father contributes no mitochondria. And this can actually be seen in plants as well, because in plants, chloroplasts are inherited maternally. So whatever plant provides the egg cell for a seed is going to be providing the mitochondria. So let's say you had, the chloroplasts. So let's say you had chloroplasts that had a mutation where they didn't produce green pigment, and they were found in the mother plant, let's say. Any offspring of that plant would also have white chloroplasts because they all inherit their chloroplasts from the mother. On the other hand, let's say you had kind of wild type normal plant with green chloroplasts, mate with any male plant, and you are going to get the green chloroplasts of the mother. And that is the kind of most important thing to look at. So now let's look at how the environment affects the expression of genes, because you can get the exact same genes as another person, for example, if you're an identical twin, but that doesn't necessarily mean you're going to be 100% identical. Environment does play a role on how your traits are expressed. A big one is, we already looked at in class, human weight, but human height has a number of different genes that contribute towards the height. So you've got your genetic potential for height. About 80% of what determines your height is due to genetic factors, and there are a number of genes that are involved. There's as many as, the latest estimate I saw was that there were about 180 different genes that can affect the height that you will become. Some of these genes may have very large effects. So for example, this FGFR3 gene, if you have a certain mutation in it, will cause achondroplastic dwarfism, which will give the person who has it very short height. And then there may be others where a mutation will just maybe give you a small variation in height. But there are also a lot of environmental factors that are important too. Very important are the health of a mother during pregnancy, as well as a lot of factors that are in play during childhood. So if a mother smokes, if she drinks, if she's exposed to anything hazardous, if she has poor nutrition during pregnancy, that can cause a change in what height the child will achieve. Similarly, nutrition, any kind of diseases, or the general health of a child, their access to healthcare, how active they are, all of those things can also have an effect on the height of an individual. And this has actually partially been seen if you look at graphs of the average height of people in the world over, the past century or two. You'll see that that average height has been growing, not because we're getting taller, but because more people have better nutrition and better access to healthcare, and therefore they're able to reach more of their genetic potential for height. Some different examples that are a little showier. Hydrangeas are a type of flowering plant, and depending on the pH of the soil that you grow them in, they will have a flower color ranging anywhere from a very bright blue to a very deep pink. And you can actually graph this. So this is a graph showing the micrograms of aluminum per gram of fresh sepal, which is basically the petals, and it shows you the wavelength of light that's absorbed, and it basically reflects these different colors that you see. So the way this works is soil pH affects how these flowers can pick up aluminum, and that affects their pigmentation. There's also some seasonal effects. So, for example, what you see here in these pictures, these two could be the same organism, and these two could be the same organism. However, their physical characteristics change depending on the time of year. So you can have these seasonal changes in appearance. This has to do with camouflage. It's an adaptation. If you are all white in the snow, you camouflage well and can hide from predators. However, if you are all white during the summer or spring when things are green or brown, you will not blend in well and you will be picked off. So it's a very important adaptation for these animals to be able to change their coat color. What happens is there are triggers from changes in either the length of a day or changes in the average temperature that can affect hormones that are produced in the animal, which will affect the color of the fur that's produced. Another interesting thing is in certain reptile species, the gender of the babies that are hatched from an egg depends on temperature. So if you have high temperature, it may mean that the eggs will only hatch males, and if you have a low temperature, that means that the eggs may only hatch females. It differs by species. So what you see here in this diagram are 3 different species, each of which produces a different percentage of males depending on the temperature. There are sometimes some other characteristics that can have an effect, but by far the biggest one I've seen the most examples of is temperature. You can also have environmental effects on other traits. So for example, when you have skin cells and they're exposed to UV radiation, that actually can upregulate the genes that produce melanin, the pigment that helps protect your cells from UV radiation. And then when more melanin is produced, it will then be transferred from these pigment-bearing cells called melanocytes into your skin cells to help protect you against damage. And then yeast mating types are kind of when diploid yeast cells undergo stress, they will undergo meiosis, and then they can mate with yeast cells of the opposite mating type, which are A or alpha. And then that allows them to recombine genes, which will hopefully produce a combination of genes that will allow the yeast to survive to a next generation. All right, that is the end of these notes. Get ready for DNA talk.